Hi, I'm Russ with Thermo Scientific, here today to talk to you about our services for developing expression cell lines. Now at Thermo Fisher, we're unique as a service provider in that we also spend over $1 billion a year on the development of next-gen media, reagents, and instrumentation that's already used at the bench for life science research. Our service scientists are able to tap into this R&D network to leverage those innovations to help tackle the most difficult biology you can throw at us. This includes making new workflows with those tools and also leveraging them, cutting-edge innovations, working with support on those R&D scientists to help tackle your most difficult problems. We're also able to provide a dedicated project manager with everything we do. We understand that outsourcing challenging biology, the important thing is to make sure that there's good communication and constant updates. Now, to tackle your cell line development needs, we have two options. One is our traditional cell line development. This is going to allow you to work in any cell background of your choice and with a delivery method of lenti or plasmid. We also have other technologies like our Jump-In and Flp-In cell lines. These are generated with a defined genomic locus where we're going to insert your gene, a safe harbor site. So we're able to generate an isogenic pool in as little as 5 weeks. The challenge here is that there's some limited parentals available, but these are in workhorse lines that are going to be great for drug discovery and research. So a quick overview of Jump-In and Flp-In if you're not familiar with this. These are pre-engineered lines that have, like I said, a safe harbor locus that is going to turn on an antibiotic selection marker and allow you to have a stable isogenic pool in as little as 5 weeks. You can also accept very large inserts in the Jump-In system, so you have a lot of flexibility even looking at multi-gene size inserts. Now, how the Jump-In technology breaks down is essentially on the left you can see you would co-transfect your host cell line with our gene of interest, flanked by the integrase or recombinase sites, and a second plasmid expressing that protein that's going to do the cutting and annealing back into the safe harbor site. And then once we see integration of that event, we're going to turn on that antibiotic selection marker, and we can easily use BLAST or Hydro and get isogenic pools in as little as 3 weeks. And what's really cool about this technology is that if you're working with multiple different cell lines, multiple different proteins, you can look at wild type and 7 disease proteins, look at 8 different lines and know they are isogenically identical with the exception of that one mutation on that protein. So unparalleled control in your ability to develop expression cell lines. We also have the ability to work with inducible promoters if you have toxic or proteins that you want potentially at a lower expression level. So looking at the larger expression workflow for traditional cell line development, we started at the beginning. We can work with your gene of interest. So you can send us the sequence file and we can synthesize those and clone them into whatever vector, whether it's an overexpression vector, a lenti delivery mechanism, or the Jump-In systems. You can send us the cell lines or we can acquire them on your behalf. From there, we do the transduction. Like I mentioned, in Jump-In, Flp-In, you're going to get integration of your gene of interest in that safe harbor locus, very defined, a single copy, whereas with traditional, we're going to get random integration, multiple copies in a variety of loci. So when we move to the stable pool generation with Jump-In and Flp-In, we're essentially done. We're going to apply that selection pressure, and then what's going to come out is that isogenic pool we'll further characterize and deliver to you. With a traditional cell line, we're going to add selection pressure again, just to reduce down the population to those cells that are expressing the gene of interest, again, in a variety of loci and at multiple copies. The next step there is to take it a step further and give you exactly what you're looking for in a clonal line where we can do limited dilution cloning or FACS sorting to make sure that we get a single population of cells and that the expression levels and biology is exactly what you're looking for and what we've defined. And from there, we then would deliver that cell line or provide any additional downstream assay development, QCs, or anything you would desire. That's how we would tackle your traditional overexpression cell lines. Again, we can do almost any edit in any cell line. If you can think about difficult biology, we're willing to talk about it, and we'd love to have that conversation. So please reach out to your commercial partner, account manager, or your cell biology technical specialist today to learn how we can quickly engineer some cell lines for you.